It's an absolute pleasure for me to join you in this 2023 annual Saudi Hematology Congress. I'd like to thank the Congress Scientific Committee for inviting me to be part of this Congress this year. I was telling someone here that this is my second visit to your beautiful city. We always talk about pre-COVID. I was here pre-COVID. And I'm here post-COVID. And my brief today is to share some of the strategies for hemophilia patients who fail gene therapy. As most of you are aware that gene therapy is with us. In fact, you'll see in the data that I'm presenting later that there are 4 patients that have been treated with commercial gene therapies. This is in addition to just under 300 patients that have been treated to date. These are my conflict of interest disclosures, reflecting largely my interface with the role players in the clinical trial space where I spend most of my life. In today's agenda, I will perhaps remind you why hemophilia is an attractive option for gene therapy. Then I'll give you an update on gene therapy programs that are currently continuing. And then I will share with you the strategies for patients who fail gene therapy screening. And I'll end this presentation with some concluding remarks. Perhaps a biggest reminder is that we've seen this unprecedented evolution of therapies in hemophilia, starting with blood and blood products. When DNA technology became available in the 1980s, we moved on to recombinant clotting factor concentrates, and more recently, recombinant clotting factor concentrates with extended half-life. The replacement therapy, of course, is not without any limitations. I've listed here only 3 of those limitations, immunogenicity being one of the biggest limitations. As you know, once you've developed inhibitors, you may well be treated with bypassing agent. And our strategies currently with the options for inhibitor patients are very limited. Sometimes the bypassing agents do not work, or the immune tolerance induction, in fact, doesn't work as well as one expects. The second limitation is the burden of treatment associated with replacement therapy. We do know that they have to be given intravenously, and that imposes a huge treatment burden to our patients. And of course, the last one is intrinsic in the pharmacokinetics of replacement therapy. The peaks and troughs result in areas of vulnerability for patients where the troughs result in breakthrough bleeds. And that is inevitable, as evidenced by many avenues, including the finding of hemosiderin deposit in the joints of these patients. It is in that context that gene therapy has actually been developed. We hope that, in fact, gene therapy will be able to address the unmet needs in these patients. And I've summarized here the AAV-based hemophilia gene therapy currently in phase 3. As you will know, there are just over 30 programs in various stages of development, phase 1, phase 2. I'm only going to focus on those programs here that are currently in phase 3. There are 2 hemophilia A gene programs in phase 3, valoctocogene roxapavovec and giroctocogene fitelpavovec, both of which, in fact, now are in quite advanced stages. And I'll take you through some of the developments in those. For hemophilia B, is tranacogene desapavovec and fidanacogene elapavovec, both of which are also quite advanced in their development. Just to remind you that they are sponsored by different companies, and if you want more details on eligibility, which we're going to talk about later here, and I'd urge you to perhaps look at clinicaltrials.gov. Perhaps I want to point out here that we're not using, in terms of the genes, the normal genes as we know them in replacement therapy. In the context of hemophilia A, we're using the B domain deleted factor 8. As you would recall, it is the B domain deleted factor 8 that can fit into the vehicle, the AAV, which can only take a certain amount of cargo. In the context of hemophilia B, we're using the Padua variant, which is the hyperfunctional variant of factor 9. And perhaps the reason I'm pointing this out, there is a difference post-gene therapy in how these two transgenes are managing. Perhaps importantly, they all use AAV, mostly AAV5, but also AAV2, AAV6, and AAV8. And I'll show you some of the data of the antibodies directed against these various naturally occurring AAV serotypes. The dosing regimens, they range from 5 times 10 to the 11 to 6 times 10 to the 13 in the valactocogene roxaparvovec program. And perhaps not surprisingly, there's been a lot of enthusiasm. Just under 300 patients, in fact, have been treated with these 4 programs, with the largest cohort, of course, in the valactocogene roxaparvovec. They've only been tried in the adult setting, 18 years and above. We don't have, as yet, gene therapy in the pediatric setting that is coming soon. And of course, we do know that some of these have been approved by regulatory authorities around the world. As you can see here, that valactocogene and etracogene have been approved by the FDA, the EMA, and we've got, at least as of last week, 4 commercially dosed patients. One from Germany with Roctavian, which is valactocogene roxaparvovec, and 3 from the US with Hemgenix, which is the etranacogene dezaparvovec. And perhaps more importantly, and I'll put this data into context as to why so few patients have been dosed in the context of commercially available gene therapy. Let's just remind you of the importance of the outcomes. In hemophilia B, we've seen very impressive rise in factor 9 activity. This is the data from Hemgenix showing that following a single infusion with this gene, you can see a steady state factor 9 activity over time, going out to 24 months, at least in this particular data set. We do know that these patients are now approaching the 3rd year. And that is particularly important to keep in mind because we're using the Padua variant of the factor 9 here, and that may explain why one has a steady state approach. Of course, the factor 9 gene much smaller than the factor 8 gene. That is probably another explanation as to why the pharmacokinetic of factor 9 post Hemgenix looks differently for hemophilia B compared to A. What is particularly important with the hemophilia B gene therapy is that patients have been dosed that have got pre-existing neutralizing antibodies against AAV, and that data is shown here. Beg your pardon. You can see here that if one looks at the entire analysis set, there is a 64% reduction in annualized bleed rate. If one looks at only those patients that were neutralizing antibody negative, there was a 72% reduction, and if one will look, 79% reduction, and if one were to look at those with positive for neutralizing antibody, it came down to 61%. And we can talk about as to why is there a difference between those who were positive compared to those who were negative. But nevertheless, the drug is approved for both negative and positive individuals. This is the proportion of individuals that required factor 9 replacement therapy, 96% reduction in the utilization of factor 9. Just want to switch over now to, I beg your pardon there. Factor 8 is slightly different from factor 9. This is data that we presented recently at EHET and ISTH, you can see that the data goes out to at least 3 years. In fact, some of our patients have actually gone up to 4 years. And the data shows that, in fact, it's a triphasic approach. There is a rise following the infusion of the factor VIII, and then a rapid drop, and then a very slow drop in the factor VIII activity. This is all chromogenic assay. Factor VIII activity is not one stage. The data is slightly different from a one-stage assay. And perhaps more importantly, one can see that there is actually a drop, compared to factor IX. And for the reasons that I've already mentioned, one needs to keep that in mind. What about the factor VIII utilization following gene therapy? We were very much impressed. There was a 98% reduction in both the factor VIII utilization and the infusion rates in the patients at the end of year 2. We've got data that now shows exactly the same figures at the end of the year 3. Now, what is important perhaps, and something that I'm going to talk about for the rest of this talk, is the key eligibility criteria in these AAV-based hemophilia gene therapies. You will note that they're more or less the same across the 4 programs. Patients with inhibitors are excluded. Patients with anti-AAV antibodies are also excluded. And patients with any liver pathology that is currently active are also excluded, and of course those with HIV. The exception, of course, is the patients who are currently considered for hemophilia B gene therapy, who may have anti-AAV antibodies. They are currently not completely excluded, as you can see in the schema there. And I want to talk mainly about the first 2 exclusion criteria and what mitigating measures are in place. There's no doubt in my mind that there's a lot one can do with patients who are HIV positive, and they are not completely excluded as long as the disease is controlled. And one obviously does a large number of imaging studies to evaluate if the liver is capable of carrying the gene, the transgene that one is infusing. So I'm not going to talk about the last 2, but I'm going to talk about the first 2. Just to remind you that we do have a fair number of options for managing patients with inhibitors at our disposal. These are the regimens that we use for immune tolerance induction. The Bond regimen has got a success rate of 100%, and the Dutch regimen 87%, and the Malmo regimen 63%. Overall, if one were to take all of this together, we're thinking about 60 to 70% of our patients may well have the opportunity to have the inhibitor eradicated, essentially making them eligible to be able to participate in gene therapy. And in fact, the question you may ask, well, what about the 35 to 40% of individuals where you cannot eradicate the inhibitor? In that case, in fact, there is light at the end of the tunnel. David Wilcox in the US has been working on this for the last 3 decades. And in fact, his persistence and patience is beginning to bear some fruit. And I'll urge you to go and read some of these papers, which sound like science fiction, but they're actually becoming a reality. And the reason I'm saying they're a reality is that he started a phase 1, phase 2 study in human, where he's going to use lentiviral gene therapy in patients with hemophilia with inhibitors, and to see, in fact, if it works. The last time I spoke to him was at ISTH in July, and he has just dosed the first patient. And I would urge you to keep your eyes and ears open, because if this technology works, then the limitation of inhibitor patients goes away completely. And admittedly, we're talking about a small number of patients, the 30 to 40% that are not amenable to inhibitor eradication or any other therapy for inhibitors. What about the patients with antibodies directed against AAV? It has been a big issue, and for those of us who are not in the space, maybe let me put those into context. In our center, we did a small study in collaboration with the Dutch group where we wanted to know what the seroprevalence was in our hemophilia B patients. Shown on the left here, you can see we looked specifically at antibodies directed against AAV 1, 2, 5, 6, and 8. I mean, we were very surprised that the prevalence was very high. In fact, to the level at which we thought none of our patients could qualify for any gene therapy for that matter. Well, we're not the only ones. If you look at the graph on the left, this is a graph from the Indian group, Alok Srivastava. They are developing lentiviral gene therapy using an AAV3 vector. And they wanted to know what the prevalence was. As you can see, and I apologize, it is slightly smaller, but you can see that the prevalence was also very, very high. So in reality, in our population, we are facing a problem. And the problem is the high prevalence of anti-AAV therapies. And of course, some of us participate quite extensively in global surveys. This is one such study, and for most of you, you'll see that South Africa actually sticks out with a very, very high prevalence of anti-AAV therapy. Well, of course, you think that we cannot participate in any gene therapy with that level of prevalence. We've dosed just over 30 patients with gene therapy in our center. And in fact, I'm going to talk about the strategies that one can use in the future. The first one that we are all familiar with is plasmapheresis that can mechanically remove the anti-AAV antibodies. Plasmapheresis, it's not anything new to this audience. It's routinely used in hematology, neurology, and oncology. You remove the plasma using the machine, and you use the immunoadsorption columns to get rid of the antibody and infuse the plasma back. The good news, of course, is that it is agnostic to any immunoglobulin, essentially takes out all immunoglobulins, irrespective of what their function would be. That one hopes that, in fact, it also removes the anti-AAV antibodies. And if in one cycle, one can get as much as 80% out of the system. In high titer anti-AAV antibody, one might require a number of these cycles. So what are some of the advantages of plasmapheresis? It is available in most gene therapy centers, in most hematology centers. We use it routinely for TTP, etc. Tried and tested and effective in removing antibodies, and does not require any additional expertise when used by people who are going to do gene therapy. The downsides, of course, is you are sticking large bore needles into individuals with a bleeding diathesis, with hemophilia. It may cause patient discomfort, and sometimes patients may actually bleed extensively. The efficacy may well be unpredictable. Depending, of course, on the antibody titer, you may need to repeat the procedure a number of times. It may require a number of rounds of procedures over time, with the patient having a large bore needle stuck in their veins, or for the sake of the plasmapheresis. And perhaps the biggest limitation, I've done just over 30 gene therapies. Not a single one of our patients have been admitted to hospital. We do it as an outpatient procedure. But if you're going to do plasmapheresis, you can't send a patient home with the catheter. You probably have to look after them in the hospital so that if they do bleed, you can manage that appropriately. And of course, one has to balance the risk-benefit ratio in patients with bleeding diathesis, such as in hemophilia. The second strategy is that of an endopeptidase called imlifidase. This is an endopeptidase that is cleaving the immunoglobulin, first published in the NEJM a couple of years ago in 2019, used in the context of transplantation in oncology. And the graph there speaks for itself. If you give it as a single dose to the patients, you can see a drop in total immunoglobulin, and over time that actually recover. In fact, the recovery is very important because we do know that other than the anti-AAV antibodies, there are other antibodies that we do need. We don't want those to go away forever. The recovery is, of course, on the slow side here because the patients were given myeloablative therapies, and in fact, the subsequent studies done in non-oncology patients shows that the recovery is much, much quicker. What is shown on the right is the proof of concept that it can work with anti-AAV8 antibodies. If you give it as a single dose, shown in the two graphs there, you can see that you give the immunoglobulin and then you give the IDES, which is this endopeptidase. You can see the drop in immunoglobulin, which is statistically significant. It hasn't been tried yet in the context of human beings, but in fact it is in the process of being tried. We should see the results very, very quickly. One hopes that in fact it works at least in humans, and one can start to use that to get rid of the anti-AAV antibodies, essentially meaning that countries such as South Africa and many other countries where the anti-AAV antibody titer is very high, one may be able to use it as a once-off to reduce the antibodies and therefore be able to infuse the patient. It creates a nice window to be able to infuse the patient. In fact, yesterday I was in New York and I was speaking to someone who does a lot of preclinical studies, and he told me that all you need is 30 minutes window during which, after the infusion, you'll be able to get your transgene and the vector into the liver. You actually don't need days on end to be able to do this. I mean, if this works, essentially the story of the anti-AAV antibodies being the limitation to gene therapy would be a story that one can forget about. And I want to come back here that in fact the inhibitors and the anti-AAV are not the biggest limitations. Just to remind you that we're talking here about 4 gene therapies that are probably going to all be commercialized very soon. 2 of these are already commercialized. In fact, the biggest limitation is the fact that they cost a lot of money. It is the access. Just to give you an indication here, if one looks at, and I'm going to go back to the previous slide, if you look at the valactocogene roxapavovec, it has actually been approved for over a year, and yet there's only 1 patient who has been dosed, and that patient was actually dosed 2 weeks ago. And the biggest limitation is the cost. At some stage, one hopes that the price will come down and these drugs become commercially viable. The cost at the moment is $3.25 million just for a single dose. Obviously, it costs a lot of money. And the same, in fact, with Hemgenix. Only 3 patients have been dosed, and these were not in big academic centers. These were in small centers in the US, which could afford the treatment here. So what can you conclude from this? I think I've sort of convinced you that there are limitations to replacement therapies, and there are unmet needs, and gene therapy certainly will be able to address those unmet needs. There are currently 4 gene therapies in phase 3 human studies that are currently ongoing, and they show very promising results, both in terms of efficacy, but more importantly, acceptable safety profile. There are several strategies that are being developed to remove the barriers to gene therapies, and I've alluded to at least two of those strategies. Despite these limitations, it is amazing that, in fact, just over 300 patients have received gene therapy in hemophilia to date. And I'm sure that, in fact, many more are going to come once we start to address the question of access and price being the biggest barrier to date. With that, I would like to thank you for your attention, and I'm looking forward to the question and answer session.